Prenez place, merci. On va commencer donc 10 minutes plus tôt avec la présentation de Sophie Picq sur l'ulcère de Burley et plein d'autres choses qui sont, j'ai pas le nom, mais Ecology et évolution de la mycobactérie en Guyane française. Je vous en prie, prenez place. On commence. La parole est à toi.
Okay. Hello, everybody. My name is Sophie Picq. I am a postdoctoral researcher. I'm currently working at the Field Natural History Museum in Chicago, but up until a couple weeks ago, I was working on this project, the ecology and evolution of aquatic mycolactone-producing mycobacteria, or MPMs, in French Guiana. So before I jump in, I want to make sure to mention that this work has been a huge collaborating effort between all the institutions you can see here. And it's involved 5 different teams, 2 in France, Jean-François Guégan and Christine Chevillon, and a lot of different expertise from public health researchers, microbiologists, ichthyologists, and entomologists, a lot of different students too. And so together we're working on why there is ecological persistence of these pathogens in aquatic habitats. So one species in particular of MPM is Mycobacterium ulcerin. It causes a disease called Burley ulcer. It's a necrotizing skin disease considered a tropical neglected disease. Its prevalence is mostly in West Central Africa, but it's been reported in all the countries you see in color on this map. The first clinical signs of infection involve a little scab or a papule or nodule that, if it's left untreated, can spread to the subcutaneous fat, lead to necrosis, and then to ulceration. So ulcers can cover a big part of the body, and as you can see, this causes, a really high level of morbidity, really bad physical impacts, social impacts, psychological impacts. A lot of the people who carry the disease get stigmatized within their own communities, and so they don't seek treatment very fast. Another reason people don't seek treatment very fast is because Mycobacterium produces a lipid called mycolactone, and mycolactone actually causes the disease to be painless, but also it interferes with the immune system, and it makes the immune system less likely to respond in people who carry the disease. So as you can imagine, this is a disease that has, a high level of morbidity, and surprisingly, we don't understand the full mode of transmission yet. So we know that the conductive environment are slow-moving aquatic habitats, but we don't understand the whole range of reservoirs. We don't fully understand how it's transmitted and how it replicates. For a very long time, only Mycobacterium ulcerans was believed to produce mycolactone, but other mycolactone-producing mycobacteria, MPMs, were found in diseased frogs and diseased fish. And so it seems that M. ulcerans is likely just a subset of MPM diversity. When we look at Burley ulcer cases in French Guiana, we found there were 2 main peaks, one in Sinnamary in the 70s and one in Mana in the 1980s. And when we look at the land use changes that corresponded with these areas in these times, we see that in Mana, this corresponded with the development of rice fields on what used to be marshy mangrove forests. And then, interestingly, in Sinnamary, after the Petit Saut Dam construction in 1994, the cases substantially declined. So it seems to us that environmental changes that affect flooding and water flow explain the rise and the disappearance of these infection clusters. When we look at MPM presence in the environment, the first detection of M. ulcerans in the environment in South America was in French Guiana in 2014 in some coastal floodplains that you can see in red. And then presence upstream Sinnamary was confirmed in 2019 and in other rural sites. And so we were a little surprised that, given that the disease became less prevalent after the 80s, and given the very low population density in French Guiana, we were a little surprised that it was that easy to find M. ulcerans in about 200 environmental samples. So our hypothesis is that it is fairly common and widely distributed in French Guiana, and that its other factors like socioeconomic transmission-related or habitat modifications that trigger these cases. So the questions we wanted to address in this study were, what are the drivers behind MPM ecological persistence, and more specifically, what are the main hosts and reservoirs of MPMs? This is in bold because this is the question for which we have data to show today, but we have other questions that I'll go over after. So to look at this question, we employed community level bioassessments across 3 seasons. So we sampled in July 2021, then in the rainy season in March 2022, and then again in July 2022. We sampled in 3 watersheds, Mana, Sinemri, and Aparag, in the upper waters, middle tributaries, and the coastal lowlands. We also sampled in 11 coastal aquatic habitats that were not linked to main river channels. And at each site, we assayed aquatic plants, soil, water, aquatic invertebrates, fish, and amphibians. And we used a combination of qPCR and PCR methods to detect MPM in those samples. So this map shows you the results of the presence of environmental MPMs that correspond to the first sampling trip, so July 2021. The localities where MPMs were detected are shown as dots with a red outline. The color reflects the temperature. We measured a lot of different abiotic factors, and we're trying to map with positivity to see if we see patterns. And then the size of the dots represents the proportion of filters that were found to be positive. So, sorry. There we go. We found MPM positivity in only the water samples, not in our aquatic plants or soil samples. And MPMs were predominantly found in the coastal lowlands of the watersheds. We found no MPMs in the coastal habitats that were not linked to the watersheds, which was interesting because in previous studies these had been detected. And then only one upstream site was found to be positive for MPMs in Upper Aparag. In terms of fish sampling, so we were sampling at each site with dip nets only. We had 3 transects at each site, and we would take approximately 10 fish per transect. So this is just giving you an idea of the diversity that we found. And then we would collect each fish in a tube individually and extract the microbial mucosal microbial community. Interestingly, we only detected MPMs in 1 fish, Hyphistobrycon, in 1 locality in the lower Sinemri site. That corresponds to a site that was highly positive as well for water samples. And so this indicates to us that even though fish could possibly be a passive reservoir for this, for mycobacteria, it definitely shouldn't, we don't think it's the main reservoir. And this is in line with other studies that have been done in other countries. Fish tend to have between 2 to 5% positivity rate. So we also sampled for aquatic invertebrates. We have approximately 400 specimens per river, so I don't have MPM positivity rates to show you on this because we're still going through the taxonomic identification. So other questions that we're looking at are, how do MPMs enter new microbial communities and get established? And specifically, what is the role of mycolactone? We're interested in looking at whether mycolactone is actually a novel weapon for mycobacteria to have an ecological advantage in those microbial communities and not really a weapon to attack us as humans. So to look at the role of mycolactone on microbial communities, we deployed field microcosm studies where we had chamber slides. Some of the slides were coated with mycolactone, some were left without anything. Then we would put them in the field, harvest them 2 weeks later, look at the biofilm on those slides, and look at the microbial communities using 16S metabarcoding. And so I'm showing you guys results from a pilot study that was conducted by part of our team in 2014 in West Central Africa, and where we did see significant differences in the microbial communities between mycolactone-coated slides and control. So we're excited to see how mycolactone affects the microbial communities here in French Guiana. Another question we're really interested in is looking at the MPM and biodiversity relationship, because loss of biodiversity has been shown to exacerbate pathogen spillover in some cases, but in general, that relationship has been poorly quantified empirically. So we're looking at pathogen diversity, host diversity, and pathogen spillover. And this seems important to us, especially in a context of forest loss and emerging infectious diseases. So to quantify biodiversity in our sites, we've been employing eDNA sampling, and we've had the pleasure of collaborating with Jérôme, Muriel, and Sébastien Brasse at EDB, who have built an incredible local resource and have developed methods that are really well adapted to these rivers. So we're looking at quantifying fish and invertebrate communities using eDNA sampling. And we've done a couple samples, so we're still processing most of them, so stay tuned. And then lastly, I don't know why the things don't show up here. We also want to look at what conditions trigger disease cases. So what conditions lead from presence of the pathogen to an infection cluster. And for this, we have a new postdoc starting this month to look at creating epidemiological models of multi-host transmission of this disease to humans. He's going to incorporate all the watershed topology and abiotic and biotic factors that we've collected. So there's going to be a lot coming out of this group. We just finished our sampling, so stay tuned. Thank you for your attention. Thank you to everyone on the team and the funding sources. And if you're interested to know more about the project, we have a website and a YouTube channel and Twitter account. Thank you.
So we'll be talking about the Brucella, which is one of the pathogens that will be next online for our discussion.
Okay, so thank you. I'm honored to speak here with you because I'm a clinician, actually, a physician, and I will talk about how we identify the novel species of Brucella causing human brucellosis in French Guiana. So first, as you know, we are in South of America, and French Guiana is just in north of Brazil, and on the left side we have Suriname. Just for you to understand the context, we have several care facilities here in the territory. So we have 3 main hospitals in Cayenne, Kourou, and Saint-Laurent-du-Maroni, which are represented on the left in the picture. And we have also other healthcare centers where we can receive patients 24 hours and 7 every day. So the first patient was a Brazilian clandestine gold miner that arrived in the border between Suriname and France after leaving his city with his name here, Belém. And he lived there for 10 years. And in 2020, he developed symptoms and went to the French border to see a doctor in Maripasola, but as he was not getting better, he was transferred to Cayenne, and there we did some samples, and we found on blood culture Brucella species after 68 hours of culture. Second patient was also from Brazil. He was also working on gold miner. He was from Macapá, which is also in north of Brazil, and he was living also in the border between France and Suriname, but he was much more near the other city named here Apatu. And in 2021, but you cannot see, I don't know why. In 2021, he developed symptoms, but he had symptoms that were evolving for several months, and he lost a lot of weight. He has low back pain, and on the Amara ear, but you don't see where there is a big abscess here, and he has an infection here. So he was transferred to several hospitals, and finally went to Kourou, where we performed a punction of the abscess, and the culture was also positive with Brucella. After all, we sent the samples to the National Center of Reference for Brucella. So we wanted to know which kind and which species it was. So first, they confirmed with a Malditov analysis that it was a Brucella species for sure. And they did more analysis with the typing of Brucella isolates using multiplex PCR. And with the Brucellator multiplex PCR, we have on the right side of the picture here, the 2 samples and they were similar to three other Brucella species: Brucella suis, which is commonly causing human brucellosis, and also Notome and Microti. But if they did another multiplex PCR, which is a Swiss later multiplex PCR, they could not confirm this similarity because those two samples were different from all the other species that were analyzed here. So they did more exams, and they did a whole genome sequencing of the two isolates. And with analysis, they were able to confirm that it was for sure a Brucella-specific genetic features. So both of those isolates were Brucella. But during a phylogenetic placement, we could see that those two isolates were different from the other one, but they belong to the classical Brucella phylogenetic order. So what? Brucellosis is a zoonotic disease. So what is it? It means that this bacteria can spread between animals and humans, people. But moreover, there's a recent publication, but it's 2020, yes. And they show here that several animals can spread the disease to humans with a red arrow. This is known that it could lead to human brucellosis. Here, this brucellosis, for example, does not lead to infection in human. For those, it's not determined, so we don't know. And for those, there is a big mystery. So we know now that we have another species here, but you cannot see, sorry, which is Brucellosis amazoniensis, which is linked to human brucellosis. But as you can see, there are different kinds of animals that can carry also the bacteria and spread the disease to the human. So which animal in French Guiana can do that? And if I go back, for the two patients, I said that both patients were gold miners, so maybe there is a link. And moreover, when we speak more with the patients, the first one said that he was living in the forest for a long time and was also having contacts with those wild animals in the rainforest. So there are more questions for next study, and one is this one study that is led by Dr. Dwin about understanding more if there are other people that develops brucellosis, human brucellosis. So she is working with a Brazilian gold miner, and we have several samples, so we can study the seroprevalence of brucellosis among them. And another question, as I already said, is which is the animal reservoir of this bacteria, of these Brucella isolates. So we have several hypotheses, like peccari, capybara, etc. And this study is led by Benoît Dutoisy, working at Institut Pasteur in Guyane, and also in collaboration with Claire Monsard and Guillaume Guiraud. So we will do several analyses, especially with a very large collection sample from wild animals to understand if they carry or not this Brucella sample. Aristopius, thank you very much.
Carry on to the next talk by Amandine Guidez on the host communities, species, and genus Culex.
So good morning. So I'm here to talk to you about the host community in Culex mosquito species from the French Guiana Zoo. So mosquitoes life cycle is made up of 4 stages: eggs, then you got larvae from stage 1 to 4, then pupa, and from the pupa emerge adult mosquitoes. They mate, and female will after require the blood meal for the maturation of her eggs and for the continuing of the cycle. Blood feeding pattern is a distribution of feeds taken on different vertebrate hosts. It is specific to species. When the mosquitoes feed on a selected host, we call about a specialist feeder. In the opposite, if we have a wide range of hosts where a mosquito is fed on, it's called a generalist feeder. In the middle, you got the opportunist one, who will feed preferentially on one host, but from which wild host could be used to if the preferred host is in low abundance or absent. So this is important for identifying vectors of pathogens, because this is during a blood meal that an infected blood meal that a mosquito can transmit pathogens. There is different cycles of transmission. The urban cycles, which associate with very domestic vectors, the enzootic sylvatic cycle, which is associated with bridge vectors, and rural cycles with epizootic vectors. So in French Guiana, this is a region in the neotropic exposed to epidemics like yellow fever, dengue, chikungunya, Zika, also with Mayaro and Tonat viruses. But it's also a place of high diversity of mammals, of birds, and of mosquitoes. There is 2,042 species described in 2020. And from 45% of these mosquito species, so nearly half of them, are belonging to the Culex genus, from which there is many vectors of arboviruses. However, the females are very difficult to identify to species levels by morphology. So molecular tools have been developed based on genital ML, where it's possible to be to identify two species. So due to these difficulties, information on the female Culex species is sometimes missing. So because of the high diversity of Culex species, it is inducing a close association with some hosts and the source of highest number of specialist species. So a collection of samples was done in the zoo site, which contain local captive species where number and localization is known, and from which potential host it's always available. So ungarded females were collected during 1 year in 32 collection ponds, mainly by Guillaume Lacour, that you can see on the right side. After this, we did a selection of the blood-fed females and goes to the identification to species level using molecular tools, especially DNA barcoding. And after that, we did pool of DNA by species and to identify the blood meal by species that we can find using metabarcoding and Illumina sequencer. So the results are a collection of more than 6,000 specimens, from which there is 64% of females. And from these females, 36% were blood fed, which is quite a high level of percentage for field work. There is a majority of Culex species found on the site, as you can see on the graph, and identification to species of selection of specimen leads to the identification of 31 Culex species only on the zoo site. And there were 5 majority species, the Culex Estor, Vaxus, Specipes, Striatus, and Denis. And from all these species, there is some vector species already known. And for example, the Culex portesi that is known to be a vector of Tonat in French Guiana. So if you look at the blood meal results, and so if we look at the species richness of the known of the zoo from the animals of the cage, and if we compare this with the species richness from the blood meal of the mosquitoes, we can see that we have quite a similar profile of mammals, birds, and reptiles, except that with the blood meal we can detect also amphibians that is not found in the cage. If we look more precisely on the species richness in common with the zoo and the blood meal, we can see that there is 75% of reptiles that are species of reptile that are in common, 24% of birds, and 56% of mammals. Now, if we look at the repetition of the species, if we if we put the repartition of species in 3 groups of blood meal, the mammals and birds which are warm blood meal, and the reptile and amphibian cold meal, then you can see that there is highest number of generalist feeder, which are the meal, and you got also in the extremity the specialist one. If we look at the mammals feeders, the majority were have preferred hosts, which were agouti, sloth, and tapir. And if you look at the human feeder of the site, there were 6 species. If we add those from the literature, we can see that the main human feeder are also the feeder that feed on a wide range of hosts. So in conclusion, we had a lot of blood-fed females with high diversity of Culex mosquito species on the zoo site. And mosquito blood meal can be, well, is known to be a useful tool to study vertebrate community. And we have found specialist feeder, but a majority of generalist and opportunist feeder, which is interesting and is the first step for the identifying potential vectors, and is crucial to assess future risk of the transmission of pathogens. Thank you.
Move on Adria Matoute presentation on parasitic diversity of the wild fauna in French Guiana.
Okay, good morning. My name is Adria Matoute. I thank the committee of the LABEX CEBA for this invitation. And today I'm going to present a study on the parasitic microbiodiversity of the wild fauna of French Guiana. So mammals are a natural reservoir for many pathogen zoonotics, such as apicomplexa with Toxoplasma, Babesia, Sarcocystis, and it can represent a risk for animals and human health. Indeed, for example, climate change can, like urbanization, deforestation, allow close exchanges between different ecosystems, and this promotes the circulation of parasites. Transmission to human can occur during very common activities like hunting or consumption of meat, especially raw or undercooked wild meat, and it can cause food hazard. There are few data on the description of the parasite in wild fauna in French Guiana. For that, TBIP initiated a project called Paralyme. Paralyme is divided in different phases. The first was to evaluate the food risk of consumption of wild meat by humans, especially for Toxoplasma. And for that, a collection has been built by volunteers. And in some, we collected average 600 samples like heart, lungs from 172 mammals. The second phase of this project was to evaluate the parasitic biodiversity in mammals and the ecological risk of the animal during my internship. This project was possible thanks to the feeder funding. So during my internship, we evaluate the rate of the animal carrying the apicomplexa. For that, we realize a sampling of the animal of the collection. We define that 64.6% of the animal studied are positive to apicomplexa. And the second axis of this internship was to compare the molecular technique. For that, we select 13 positive samples. We realize Sanger sequencing, new generation sequencing. Our team realized the nanopore sequencing, and we sent the same sample to a society called Genoscreen, and they realized the Illumina sequencing. We defined that the new generation sequencing is the most appropriate tools to reveal the parasitic diversity and the protozoan co-infection. So the next stage is the Now the next stage is to deploy the new generation sequencing on the sample of the collection, but now with another protocol, because during my internship, we use the protocol set up in the hospital because we work with them. So during this stage, we realize a screening of the PCR of the DNA of the samples. This stage is still in progress. We target a conserved gene called small subunit ribosomal DNA to target the phylum of Apicomplexa. We realize a library and we're sequencing with the nanopore technology. After sequencing, we obtain raw data. So we realize a pre-processing to demultiplexing, trimming, barcode, and we discard reads shorter than 600 base pairs. Then we realize an alignment to SILVA database at 90%. At this step, each read is assigned to one OTU. One OTU corresponds to a cluster of similar sequences. At the end, we make a visualization of the taxonomy and we study the diversity. So we define that 70% of the individuals studied are positive to Apicomplexa. The animals studied are listed in the table. And we're sequencing 61 samples with the two protocols. Then we perform a taxonomy assignment of the reads with SILVA database. An OTU table has been built, and we select the highest number of reads that's not exclude many samples, and we carry out a rarefaction curve, and we observe that at 1,000 reads for the sequencing depth that we reach the plateau. So we're sequencing enough, and the parasitic diversity present is exhaustive. For the taxonomy visualization, as you can see in green, we have many unassigned reads. So there are many sequencing that are not referenced in SILVA database. After bioinformatic filtering, we can affirm the presence of the presence of the parasite in 21 sample among the 61 sample sequencing due to the high error rate of the technique. Another limit of the new generation sequencing is that we can describe to the gender only. We reveal an abundant parasitic microbiodiversity. For example, we observe Theileria and Cytozoon in Dasyprocta. We observe Toxoplasma and Sarcocystis in Tamandra. We reveal co-infection in 10 samples among the 21, and there is the presence of potential pathogen genera. There are few studies concerning the parasitic biodiversity in biopsy in Amazonian area, especially with new generation sequencing. With unassigned reads, we realize a comparison with another database, which is NCBI, and we obtain similar results. So we conclude that the database are not sufficiently referenced. Due to the error rate of the technique, we can select a lower percentage of identity when we realize the alignment of the read with the database. For example, we can choose 80%. This percentage is less restrictive, but we reduce the unassigned number and we can increase the number of samples after filtering. To conclude, the presence of parasitic biodiversity. To conclude, we reveal the presence of parasitic diversity and co-infection. We reveal the presence of potential genera, pathogenic genera such as Sarcocystis, Toxoplasma. The food risk is present. There is a presence of potential ecological risk because species of animal can be decimated. There is a study. It is innovative. It is an innovative study because we work on a tissue matrix and we combine that with new generation sequencing. We implement a parasite sequencing technique locally. We're going to determine the species of the pathogenic genera with complementary tools like Sanger sequencing. We would like to increment the database with South America data, and we're going to transpose the technique to other issues. Thanks for your attention. Thanks to my team and all people who contribute on this work.
So we're welcoming now Sourakhata Tirera, who is from Institut Pasteur Guyane, and will be talking about the viral diversity of neotropical rodent hosts.
Hello, everyone. I'm going to present to you my work about the influence of habitats on viral diversity in neotropical rodent hosts. This work was published in various journals. In French Guiana, rodentia present a great diversity of species with 36 known rodent species. The most specious family of rodents is the Chrysetidae family with 19 species, followed by Echinidae family with 7 species. These rodents are known to host many zoonotic viruses, with hantaviruses causing pulmonary syndromes, and arenaviruses with the patavirus having a pathogenicity not well established, and LCMV, which is a cosmopolite pathogen linked to Mus musculus. The habitats in which you found these rodents is almost all the type of habitats with pristine forests, savanna, agricultural, very urban and urban areas. Despite this data, we have not so much knowledge about the ecology of the rodents linked to the viral diversity and viral pathogens and how and the drivers of this viral diversity. So our question was, if the habitat type is a driver of viral diversity. So our sampling goes so on with 7 species sampled in 4 types of habitats. We had from the Chrysetidae family, Ocomis genus with Ocomis orientepui and Ocomis bicolor, and only 1 species in the Zygodontomis genus with Zygodontomis brevicola and Elaemis genus with Elaemis youngonus and Elaemis megacephalus. On the other hand, at the Echinidae family, we had 2 species from the genus Proechimis with Proechimis cuveri and Proechimis guyanensis. All these 7 species were sampled in disturbed forest habitats. Zygodontomis brevicola were present in 2 more habitats with savanna and periurban. In pristine forests, 3 species were sampled, namely Elaemis megacephalus and the 2 species of Proechimis. This gave us 12 species habitat combinations with 187 individuals. For all individuals, we sampled kidney, spleen, and sera, and extraction was made before amplification, and Illumina sequencing were done with 36 pools of organ, species, and habitat. Our bioinformatic pipeline works with clean reads as de novo assembled to get contigs, which are compared to reference databases to make taxonomic assignment. At this step, we get organisms identified. After that, we did a second part of analysis with mapping the reads to the contigs. This gives us the number of reads per contig, and the contigs linked to organism made quantification of number of reads by organism. So we filtered at 10 reads to get a consolidated data set. We measured diversity with 3 types of metrics: the richness of species, which is the number of viral species; the abundance with the quantity, the number of individuals representing the number of individuals in a population; and equitability and alpha diversity, which is the relationship between species richness and abundance. We used Rennie's entropy to represent this alpha diversity with a formula that gives us a recovery of richness with alpha value equal to 0, Shannon with alpha value equal to 1, and Simpson with alpha value equal to 2. These metrics have an advantage to give a weight to the rare species in communities. And the interpretation of Rennie's entropy and with the Hill numbers goes so on. A community A is more diverse than a community B if for all values of alpha, we have higher values for community A than B. So after sequencing, we obtained 1 billion reads and 5 million contigs. And viruses represented 15% of contigs and 8% of reads. So the taxonomic assignment of viruses for the 12 species habitats gave us 27 viral families. And we can observe in the heat map here that there is an unequal distribution of viral families according to species habitats that are represented here. So we continue our analysis by going to viral general level and only considering species that were sampled in more than one habitat. This gave us a combination of 9 species habitats and more than 60 million of reads. And we made a matrix with viral genera per species habitat and the number of reads linked to each genera. And the results of alpha diversity analysis gave us for Proechimis guyanensis an alpha diversity more important in pristine forest and less important in disturbed forest. We have the same tendency for Proechimis cuvieri despite less clear. And Hylaemis megacephalus also gave us this same tendency with a profile less clear too. And Zygodontomis brevicoda sampled in 3 types of habitat showed more important diversity in savanna habitat, followed by disturbed forest, and the least important diversity were found in peri-urban areas. After that, we tried to give insight about the rare species, which are the less abundant ones, by derating them and recalculating the richness of species and measuring the loss of richness between the initial values and the new values. So by doing so, we observe that more richness were lost in pristine forest and savanna, meaning that for this species, we had more rare species in the most diverse habitats. And there was an exception with Hylaemis megacephalus with higher value of loss of richness in disturbed forest. We observed that for Proechimis cuvieri and Hylaemis megacephalus, there were many taxa of insects in disturbed habitats, maybe explaining their profile of diversity kind of different from the other species. And not shown here, but in Proechimis cuvieri, we found potential arboviral species, and we did some phylogenetic analysis, and this was close to species found in Brazil and in the region. In discussion, many diversity differences are explained by presence of rare species in pristine habitats and many dominant species in disturbed habitats. This was a pattern of diversity found also in marine metagenomics data, where rare bacterial species had high richness and maybe a high impact on global biological processes. On the other hand, looking at Hylaemis megacephalus, the kind of virus found in these samples may be present with adaptive jet of this species, as it was shown also in other virums in New York City for Mus musculus and Rattus norvegicus, and this kind of pattern was only found in disturbed habitats. The same for Proechimis cuvieri, with blood-feeding mosquitoes that may feed on less abundant, less diverse mammal species, and this may favor the transmission of arboviral species to humans. So the variety of viral pathogens may slow the diffusion through two concepts: the dilution effects with the presence of non-efficient hosts, and the strict condition of the virus species themselves. And this may, the strict condition for them to diffuse may reduce their diffusion. There is maybe a reduced emergence risk in French Guiana, such as we have ecosystems that are preserved, and we have low population densities, lowering the risk of transmission. But this situation may evolve, such as the anthropogenic pressure are going to grow and situation may change. So thank you for your attention and all partners.